Good morning, children. And now we have to study the dark reaction, as we have completed with light reaction, that is non-cyclic as well as cyclic, and we have also studied the chemo-osmotic hypothesis, that is how the formation of ATP occur in that process. Now the product which is formed in light reaction is used in dark reaction. So how it is used and what is dark reaction, that we have to study now. Dark reaction is also known as biosynthetic reaction or biosynthetic phase because it involves the formation of glucose, or it is also called as biosynthetic pathway. It was first studied by scientists, that is Melvin Calvin. So it is also known as Calvin's cycle, and Melvin Calvin studied this cycle in a green alga, and he actually exposed the green alga to the sunlight, and after some time it will take out the alga and make them die out, and after that they will do the chemical reaction to find out what is happening next. So by this process Calvin do this experiment, and for doing experiment he used carbon 14. As you all know that carbon 14 is a radioactive carbon, so he used that, and when he studied, he found that the first compound which is formed and which is a stable compound which is formed is 3-carbon compound. 3-carbon compound is formed during the process, and this 3-carbon compound which is formed is phosphoglycerate or phosphoglyceric acid. It is phosphoglyceric acid which is formed during the process. But previously it was saying that the 3-carbon compound is formed as 1 carbon come from carbon dioxide, and in plant there was 2-carbon compound were present that will combine to form a 3-carbon compound. But actually this not happen. When the study goes on, it was the inference come that there were 5-carbon compound which is present in plant, and this is RUBP, that is rubulose biphosphate. It is rubulose biphosphate. This is the 5-carbon compound which is present there, and with the help of this the 3-carbon compound is formed. Now for the formation of PGA, it requires an enzyme also, and that enzyme is called rubisco, that is rubulose biphosphate carboxylase oxygenase. This is the rubulose carboxylase oxygenase is the enzyme which is required for the process of carboxylation. So actually this C3 cycle occur in 3 steps. The first step of cycle is carboxylation. The second step is reduction, and third step is regeneration. So these are the 3 steps that occur in Calvin cycle, and the formation of this 3-carbon compound or 1 molecule of glucose will take place. Now we have to study how this cycle go on. Now student, these are the 3 steps, that is carboxylation, reduction and regeneration, that occur in a Calvin cycle. Now what happened in Calvin cycle, we are going to study that. The carbon dioxide that is present in atmosphere, okay, it will be taken up by the Rubisco enzyme. It will get mixed with a RUBP in a presence of enzyme that is Rubisco. Okay. Rubisco. Okay. And to explain this cycle, as such, from atmosphere only one molecule of carbon dioxide is taken by one molecule of RUBP. But to understand the cycle more precisely, we are taking 3 molecule of carbon dioxide from the atmosphere and it is mixed with 3 molecule of RUBP from to the carbon dioxide. Now, in carbon dioxide only 1 carbon is there, but in Rubisco there was 5 carbon is there. In RUBP there was 5 carbon is there. So here we have 15 carbon. Okay, because we are taking 3 molecule of it. Why we are taking 3 molecule, I will explain it later on. Okay, and we are taking 3 molecule of carbon dioxide. Now it will mix, carbon dioxide going to mix with RUBP in a presence of enzyme Rubisco, and they form a 3 carbon compound which is stable in nature. And the 3 carbon compound which is first formed, a stable compound that is formed is phosphoglycerate. Phosphoglycerate is found. It is also pronounced as PGA. It is also called PGA. And as we are taking 3 and 3 molecule from here and 3 molecule of here, so here it is found 6 molecule of phosphoglycerate. Okay, 6 molecule of phosphoglycerate is formed, and this is as 3 carbon compound. So in this we have 18 carbon here. Okay, we have 18 carbon here. Now the next step that occur in cycle is that by utilizing energy, the phosphoglycerate undergo the process of phosphorylation, and now they will utilize actually and for phosphorylation they need energy. So as there are 6 phosphoglycerates, so we need 6 ATP that will ultimately dissociated into 6 ADP plus 6 IP. That is, inorganic phosphate get dissociated. Okay, and this 6 phosphoglycerate, now it will get converted into 1,3-diphosphoglycerate. It is converted into 1,3-diphosphoglycerate. And as here is also 6 molecule, here it is 6 molecule. So here are also 6 molecule of 1,3-diphosphoglycerate is formed. Now, so 6 molecule means it is now having 18 carbon. Okay, because 1 is having 3 carbon, so 6 is having 18 carbon. Now, this is the process of carboxylation. Till here the carboxylation occur, and now the next process, that is reduction, is going on. So for reduction we have to use the compound that is formed in non-cyclic reaction, and that is the reducing agent they have formed, and the reducing agent they have formed is NADPH2. NADPH2 is the reducing agent that is formed, and this NADPH2 get reduced to NADP plus IP, 6IP. Okay, 6IP will join it. 6IP will going to join it, which is present here. And by this diphosphoglycerate undergo reduction and it will convert into aldehyde. And the product which is formed is PGAL. The product which is formed is PGAL. PGAL means phosphoglyceraldehyde. It is phosphoglyceraldehyde is formed. Now here it is also a 3-carbon compound. It is also a 3-carbon compound. It is also a 3-carbon compound. So 3-carbon compound is formed. So it means that 6 molecule of PGA is formed. From 6 molecule of 1,3-diphosphoglycerate, 6 molecule of PGAL is formed. Now what happened? From here, 1 molecule of PGAL will come out, whereas the 5 molecule will go on in a cycle. 5 molecule will go in a cycle, but 1 molecule will come out. 1 molecule of PGA will come out, and that will help in a formation of glucose. That will help in a formation of glucose. As 1 PGL is having 3 carbons, so 2 PGL is required to form glucose. Now what happened? Till here the reduction occurred. Now the regeneration of RUBP will go on. That is the third step. So what happened? This PGAL now undergo the process of phosphorylation, and this is phosphoglyceric acid. From here 2IP will reduce and it will converted into Rubisco 5-phosphate. Okay, it will convert into Rubisco 5-phosphate, and from Rubisco 5-phosphate, here again what happened? ATP will convert into ADP, and from 5-phosphate this will convert into RUBP, that is Rubisco, that is rubulose phosphate. So this is the whole process that will go on. Now the question arises, why we have taken 3 carbon? Actually it is easy for convey you that 1 molecule I have taken here and 5 molecule go in a reaction in the process of regeneration of RUBP. If I have taken 1 molecule, then we have to say that 1 by 6, that is 1 fraction will come in a formation of glucose, and other 5 fraction will go into the cycle, and that will be difficult to calculate and that will be difficult for you to understand. That's why I have taken 3 carbon and this cycle will go on. Now, in nature it does not occur so. In nature 1 carbon will enter and then they'll form, then again the second carbon enter into the Calvin cycle, and then the third carbon, 4th and 5th and 6th and so on. So 6 Calvin cycle is required to synthesize 1 glucose molecule. So 6 Calvin cycle is required to form 1 glucose molecule, but I have shown here as 3 glucose molecules so that you can understand it better. Now the thing that is required, that what is used in it. So actually this is 3 carbon compound, so it means that 2 such cycle is required to synthesize 1 glucose molecule, and in 1 cycle 6 ATP are utilized there. Here 6 ATP are utilized. So means in 2 cycle 18 ATP will get utilized. And if we talk about NADP, so here 6 NADP H2 will utilize. So if 2 cycle is there, then 12 NADPH2 is required in the cycle. So ultimately we require these two things for the C3 cycle, that is 18 ATP and 12 NADPH molecule. This much is required to complete or in a formation of one glucose molecule. So this is the Calvin cycle and it will occur in 3 steps, that is carboxylation, reduction and regeneration. When carbon forms ultimately 1,3-diphosphoglycerate, it is the process of carboxylation. Then 1,3-diphosphoglycerate converted into aldehyde, that is phosphoglyceraldehyde, it is the reduction. And again from this phosphoglyceraldehyde, the formation of RuBP takes